Hello, I'm Adele Schmicklermann.
I'm Alexander Fastner.
And we are students at Frost Lab at the Technical University in Munich. Today we're going to present to you our project, Protein Embeddings to Phylogeny, Exploring Evolution in Space. This project was supervised by Kyra Erckert, Tobias Oleni, Tobias Senona, Ivan Kolodadov, and Burkhard Trost. For our project, we wanted to construct phylogenetic trees directly from protein embeddings. To do so, we first needed to figure out whether protein embeddings contain any kind of evolutionary information. So we took proteins from unit class 30 and unit class 90 and took the sequence similarities and correlated them with embedding dimensions. And indeed, we found that some dimensions of the protein embeddings correlate with the sequence similarity of the proteins. So we took the Prot5 and ISMB embeddings, applied dimension reduction techniques such as variational autoencoder, t-SNE, and PCA, since we found that some dimensions of the embeddings correlated more than others, and then computed the distances with Euclidean, Manhattan, cosine, and triangle area similarity and sector area similarity. And then clustered those distance matrices with neighbor joining and QPGMI and constructed the phylogenetic trees from those.
For our project, we used several data sets, the phosphatase dataset, kinase dataset, but shown here on our poster are the results from our KLK, or Callicrine dataset. What we see are the neighbor joining trees that we finally constructed in both circular and non-circular form. What you see in the top right is the reference we are comparing to, which is a tree constructed using MSA, or multiple sequence alignment technique. For our dataset, shown in bright green is the outgroup we have, which will be important later. We concluded at the end that we need many metrics to see how these trees can be compared to each other. And we tried Robinson's fold, symmetric distance, Euclidean distance, and the changes in the partitions of the branches. But finally, we realized upon visual inspection that very similar trees have a big range in the resulting values of these metrics, and that these were not great tools to discover which tree was closest to the reference tree. So in conclusion, we use visual inspection and prior knowledge to compare these to our reference tree. In the bottom left-hand corner, you can see the results from our VAE. In the right, you can see the results from t-SNE. We also tried PCA, but t-SNE performed better than PCA in almost every aspect. In the top right left-hand corner, you can see the non-trained tree, and you'll notice that it's, unlike the VAE and the t-SNE, manages to correctly put together the outgroup, the bright green group I was talking about earlier, as in our reference tree. Our non-trained tree is not using any dimensionality reduction techniques. It is using directly the Manhattan distance of the sequence embeddings and putting that directly into neighbor join. In conclusion for our project, we find that the non-trained tree with no prior training and no dimensionality reduction performs best for taking protein embeddings and constructing a phylogenetic tree.
If you're interested in reading more about this project, you can find a link to our GitHub with the paper and also with the scripts for this project. And if you have any other questions or any more detailed questions, feel free to reach out to us, and we're happy to answer your questions. Thank you.
Thank you.